All right, guys, welcome back to week 4 of the case studies in science class. So this is the last week on evolution. So next time you guys come, you're not going to see me. I know, we're all broken up about it. So we're going to try to wrap up this section on evolution. I'll just say that 4 weeks is not a lot of time to cover evolutionary theory. So if you're more interested in evolutionary theory, you want to learn more about it, you know there's lots of other courses and other material that you guys can hook up with, if that's your thing. If not, then hopefully I'm going to get us through week 4 and we'll be good to go. So I'm going to start with basically what we ended with last week, which was some of the sort of deep criticisms of evolutionary theory that are arguing that these criticisms of evolutionary theory are so profound that we really got to scrap the whole thing and say there's something else going on. And this comes from, you know, the Discovery Institute, the people who, you know, are really in charge of building this intellectual enterprise of intelligent design as an alternative to evolution. And we'll go through these really two criticisms of evolutionary theory over the next hour. But I'm going to start by taking a step back and really kind of looking at how the scientific endeavor works. And I'm going to start with a quote by Isaac Newton, who says, "If I have seen further into the nature of the world, it is by standing on the shoulders of giants." And of course, what he kind of meant by this was that the scientific enterprise doesn't spring forth altogether. It doesn't spring forth from the mind of one individual. It's built up over generations as researchers continue to work on the ideas of previous scientists, and we get this sort of incremental accretion of knowledge. And so I'm going to start with an analogy. And remember, I've said before, like, be careful of analogies. But I'm going to start with an analogy anyways. And it's the old building a house analogy, right? So if you set out to build a house, it proceeds in this sort of organized manner of steps. So you start with the foundation, you build up the walls, you build up the roof, you add the details. So there's this accumulation as you go. And a lot of science definitely works along these lines, and really most of the intellectual endeavors that humans have done you can think of in this way. It starts with some sort of initial ideas that build the foundation for this intellectual enterprise. So for example, in the early 1800s, some scientists thought, hey, what if species actually aren't always the same? What if they have changed in the past, and what if they continue to change in the future? How does that change how we think about life? So there are these initial ideas, and then on top of those, we add other information to sort of build on those ideas. So Darwin came along and said, look, there's all this evidence that species do change, and natural selection is a potential mechanism that would cause them to do that. And then over time, we add more new knowledge on top of the knowledge that was built on these initial ideas. So we get things like population genetics, bringing a quantitative framework into the picture, and we sort of keep doing this as we go. And so we build information on top of previous work. And so this is a really great process because it allows understanding to grow. So we can take the ideas of the past and we can build on them to accumulate more knowledge and more understanding over time. But there's a major problem to this whole sort of approach in that it's sensitive to previous errors. So if somebody made a mistake 3 generations ago and we built on top of that mistake, then everything that we built on top of it is suspect and could fall apart. So really what we want to do as scientists and as human intellectuals is to balance the power of being able to build on the prior work of others to improve our understanding and our knowledge with some capacity to revise previous understanding if we find that it was wrong. And so science does this through an iterative process of experimentation and predictive modeling. And so we'll go over sort of how that works. Now you're probably more familiar with the idea of experiment in science. An experiment is just sort of a controlled system for asking a specific question. And the better controlled and the better rigorous it is, the better you can answer a specific question. So you design an experiment to answer this question, and you get some result out of the experiment. Either this answer, or it's ambiguous, or whatever. But you get some result out of your experiment. So that result is very specific. It says, under these particular conditions, when I do this, this happens. Right? So how do we go from that sort of specific bit of information that we can get from an experiment to something more general that we can apply to understand things, to make decisions, that kind of stuff? So what we can do, and what we as scientists do in general, is we take the results from our experiments and from other experiments, and we put them together into some sort of model of how the world works. It can be some small part of the world, it can be some bigger part of the world, but it models something that we think is going on. And hopefully it's consistent with the results that we have. That would be good. But this modeling process also makes predictions about what should happen. So it should say, you know, if this model makes sense, if it's right, then if I do this, this should happen. So, and again, the more specific the predictions, the better in terms of scientific testing. Because what we ultimately want to do is take the predictions of our model and test them using experiment. So if the model predicts this should happen, I should be able to design an experiment to test that prediction. And then if that prediction turns out to be wrong when I test it experimentally, then there's obviously something wrong with the model and I need to fix it. If the predictions of the model are all accurate when I test them objectively, then the model seems to be working, at least to the extent to which I've tested it so far. So that's sort of how this process works. And you can see how you can kind of iterate around this loop and build up better and better models. So sometimes models fail. That's important. I don't know if you guys are old enough to remember, but in grade school we used to build these models of atoms, and they were based on the solar system, right? So the nucleus is like the sun, and it's in the middle, and the electrons are like planets, and they orbit around the sun, right? And that's how we try to understand atoms in grade school. Now, we know that that's wrong, and we can show through experiment that that doesn't make sense, and then we can revise that model to what we have more or less today, which is a quantum mechanical model of how the atom works. So sometimes models can make what seem to be outrageous predictions that turn out to be accurate. So part of Einstein's theories of relativity predicted that there should be these gravitational waves. Don't ask me what they are. I'm not a physicist. But they've now been confirmed through observation and experiment. So models can help us design experiments that then either support the model or not. So right now, and you guys have seen this before, so right now, I would say that the scientific consensus is that evolution is our best model. That is, it does the best job of explaining the similarities and differences that we observe in nature. So right now, it's the model that we use. But is it right? Are there problems with evolutionary theory? Do we need to revise it in some way? And last time I talked about a bunch of sort of what I would call, open questions that scientists are beginning to think might cause us to change evolutionary theory or revise it in some way. For example, epigenetic inheritance is something that's coming on the horizon that if it turns out to be a major force, it really changes how we think about evolutionary theory or the developmental process, these kinds of things. I'm not going to focus on those too much today because these are things that fit very well within the natural world, and we could think of ways to incorporate them into existing evolutionary theory. But there are other criticisms of evolutionary theory that people have argued are really critical, that we need to, in a sense, throw out evolution and think about alternatives to evolutionary theory if these criticisms make sense. So I'm going to go into some of these criticisms and talk about the reasoning behind them and see if we can think about the reasoning and make sense of these criticisms. And the first is this idea that there's this digital code in DNA. And if you go back to lecture 3, if you want to refresh your memory about what this means, please feel free. That's why we record them. So the idea is that the genetic code is something that we would think maybe would be difficult to generate through an evolutionary process. The reasoning behind the argument kind of goes like this. The genetic code, the way that DNA controls cellular and organism behavior, is kind of like a computer program. Another analogy, just so you guys remember analogy, be careful, but the genetic code is kind of like a computer program. It's a set of instructions that are encoded in a molecule that tell the cell what to do and tell the organism what to do. Not behaviorally, but in terms of development and basic operating. So we're equating genetic code with computer code, right? So if that's true, and we look at computer programs, we see that all of the computer programs that we have were designed and implemented by a programmer or a team of programmers. Somebody with intelligence designed these computer programs. And so, because genetic code is like computer code and computer code is designed, then can't we infer that the genetic code was also probably designed, right? So that's the essential nature of this argument. And so how can we think about this argument and evaluate its strengths and weaknesses? Well, one thing we can do is we can just say, well, sure, the genetic code is kind of like a computer code in some ways, and in some ways it's not, right? So the genetic code is a molecule. It's made by chemistry. It exists in a living system. Computer code is often text-based or binary. It exists in electron fields inside a computer. There are similarities, but there are also differences. And maybe one of those differences is that one's designed and one's not. So that's one way where this argument might not make sense, and that the analogy is imperfect. Genetic code and computer codes are not equivalent. They are similar in some ways and different in other ways. And if that's true, then this argument could potentially fall down. I'm not going to take that track here today. I want to think about another aspect of this analogy, and that is the premise or the assumption that all computer programs are designed by an intelligent programmer. And I have a PhD in computer science, so I can do this, and it's kind of fun. And I'm going to ask that as a question. What's the evidence that all computer programs are designed? Or are there computer programs that aren't designed? And in fact, there are a whole class of computer algorithms that are not designed by an intelligent programmer. And these are generally classified into a group of algorithms called either machine learning or genetic algorithms. And they're called genetic algorithms because they are based on ideas taken from evolutionary theory. And basically what this programming paradigm does is it uses a model of evolution based on biological evolution to generate programs that solve complex problems without a programmer. So it's totally based on evolution, not designed by any human programmer, and yet we're able to generate these complex programs that solve problems. And some of the problems that these programs have been used to solve are very complex, like routing things, controlling those driverless cars, predicting financial markets, and things like that. These are a class of algorithms that are typically applied to problems that we can't design a solution for. If I try to write a program to predict a financial market, I might go, this is too complicated, too hard, I can't find a solution, so I'm going to let evolution do it, which is kind of interesting. We've actually used some of these in our labs to try to understand how molecules interface with one another complex problem. So these are actually used today to solve problems, and we know that they're not designed. So I'm going to show a short video that just kind of shows you an example of how some of these genetic algorithms work. And I may actually talk over it a little bit. There's no audio. There's just some music in the background. So I'll try to explain what's going on a little bit. So this is an example where we're trying to make a computer program that will control these little robots in a video game. And I apologize, it's a slightly violent video game. These two little tiny robots are trying to shoot each other. But it's the best example I could find. It's rated for all audience. So they started with this problem of these two little robots that are trying to shoot one another. And when they hit the other robot, they get points. And they start off with a random solution. And it looks pretty crappy. So the little dots are the robots, and the lines are where they're looking. And then you'll see these little black dots they're shooting, and they're trying to hit the other robot. And so initially, these robots are really bad at their task. So now they're using evolution to select robots that are slightly better at doing it. And you can see some more complex behaviors starting to evolve. Again, no design. This is a process that's completely undirected. And so over about 20 generations, you start to see things that aren't just wheeling around being stupid. They seem to be acting more towards accomplishing their goal. They're trying to, so the red one's trying to hit the green one with the ball, and the green one's trying to hit the red one, and they get points if they hit the other one. So they're trying to avoid getting hit and hit the other one. So you can see at the top it shows like their points. So now 55 generations of artificial evolution. And you start to see, for example, this red one is dodging or beginning to learn how to dodge the shot and avoid getting hit. So again, that dodging strategy is not something that a programmer put into the system. It's something that emerged through a process that's designed to mimic how we understand biological evolution. And so that was just sort of a silly example, but it's one that only takes a couple minutes. Like I said, these are used to solve more complex problems. But the point is that this argument is based on a false premise. Not all computer programs are designed. Yes, some of them are, but there's a whole class of computer programs that aren't designed, that are generated completely through this undirected evolutionary process. And so because of that, we can't infer that the genetic code has to be designed because computer programs are designed. Some of them aren't. And so it could be that perhaps some genetic codes are not designed as well. So that's what I'm going to say about this argument that the DNA code is evidence of design. The next one I'm going to talk about is perhaps a little bit more of a meaty argument. It's been around for longer. And this idea that when you look at how molecules coordinate their efforts to achieve a goal, there's this complex machine-like behavior, which doesn't seem like it could come about through an undirected evolutionary process. And in general, when we look at biological systems, they're very complex and they're integrated. And how could these complex integrated things arise through this gradual accretion of simpler things. I'm going to show, again, a short video. This is from a Nova TV show about the bacterial flagellum, and I'll just put it in a little bit of context. I think this is from 2005. There was a court hearing about whether intelligent design should be taught in biology courses in the United States as an alternative to evolutionary theory in science. And so that's sort of the context for this. But try to look past the context and think about what they're saying in terms of the way that these molecular machines may have been put together. I won't talk over this one.
A scientist and senior fellow at the Discovery Institute, Michael Behe is the author of the popular intelligent design book Darwin's Black Box, and dozens of papers unrelated to intelligent design published in peer-reviewed science journals. Behe refused multiple invitations from Nova to be interviewed for this program, though he went on record in the trial.
Dr. Behe, what is your profession?
I am a professor in the Department of Biological Sciences at Lehigh University in Bethlehem, Pennsylvania.
And you're a biochemist?
And that's correct, yes.
How long have you taught at the college level?
For 23 years.
Sir, what is intelligent design?
Intelligent design is a scientific theory that proposes that some aspects of life are best explained as the result of design, and that the strong appearance of design in life is real and not just apparent.
Is intelligent design based on any religious beliefs or convictions?
No, it isn't.
What is it based on?
It is based entirely on observable, empirical, physical evidence from nature, plus logical inferences.
Now, when you use the term design, what do you mean?
Well, I discuss this in my book, Darwin's Black Box, and a short description of design is shown in this quotation from Chapter 9. Quote, "What is design? Design is simply the purposeful arrangement of parts. When we perceive that parts have been arranged to fulfill a purpose, that's when we infer design." End quote.
Part of the defense strategy would be to show the judge examples of biological systems they claimed were too complex to have evolved by natural selection, and therefore must have been the product of a designer.
Can you give us a biochemical example of design, Dr. Behe?
Yes, that's on the next slide. I think the best, well, the most visually striking example of design is something called the bacterial flagellum. Now, this is a figure of a bacterial flagellum taken from a textbook, which is widely used in colleges and universities around the country. The bacterial flagellum is quite literally an outboard motor that bacteria use to swim. And in order to accomplish that function, it has a number of parts which are ordered to that effect. Now, this part here, which is labeled the filament, is actually the propeller of the bacterial flagellum. The motor is actually a rotary motor. And most people who see this and have the function explained to them quickly realize that these parts are ordered for a purpose and therefore bespeak design.
Under the microscope, bacteria powered by flagella seem almost acrobatic. They tumble, corkscrew, and pirouette, thanks to that whip-like filament. Driving this propeller is a tiny motor, part of a complex structure made of about 40 different kinds of proteins.
The bacterial flagellum looks like a sort of Jules Verne notion of what the future looks like. It has a strange sort of mechanical quality to it, these sort of cogs and waving tails and stuff.
And according to Behe, if any one of these parts is missing from the system, the motor can't function. Behe calls systems like this irreducibly complex, a term he coined. And he argues such systems could not have evolved naturally.
The idea is that there are certain aspects of life, perhaps organisms or organs or even cells, that in a sense could only have come about as a whole. In other words, it was very unlikely they could have come about through just a kind of contingent combination of parts over even millions or billions of years, but rather in a sense had to be created whole cloth, all together at once, because everything fits together so well that to remove one part the thing wouldn't function.
Have other scientists acknowledged these design features of the flagellum?
Yes, they have. If you could advance the next slide. In 1998, a man named David DeRosier wrote an article in the journal Cell, which is a very prestigious scientific journal, entitled The Turn of the Screw, the Bacterial Flagellar Motor. Now, David DeRosier is a professor of biology at Brandeis University in Massachusetts and has worked on the bacterial flagellar motor for most of his career. Now, in that article, he makes a statement, quote, "More so than other motors, the flagellum resembles a machine designed by a human," close quote. So David DeRosier also recognizes that the structure of the flagellum appears designed.
What I wrote was, this is a machine that looks like it was designed by a human, but that doesn't mean it was designed. That is the product of intelligent design. Indeed, this more has all the earmarks of something that arose by evolution.
Using an electron microscope, DeRosier produces ghostly pictures like this one, revealing the inner workings of what's been called the world's most efficient motor.
This is the drive shaft. This transmits this torque generated by the motor. That would then turn the propeller, which would push the bacterial cell through the fluid.
Michael Behe has argued that the flagellum could not have evolved, since its parts have no function for natural selection to act on until they are fully assembled. But evidence that refutes Behe's claim of irreducible complexity comes from a tiny syringe that injects poison, found in some of the nastiest of all bacteria.
This is a structure found, for example, in Yersinia pestis, the bacterium that causes the bubonic plague. And look at the similarities. Now, this structure doesn't rotate, but it still has to extend this structure, which is equivalent to the rod, the drive shaft here. It has to extend that because it needs this little channel. It's like sort of like a syringe. So the virulence factors that are made inside the cell, which is down here, can be exported, pushed up into this hole, and exported out through this long kind of needle, perhaps into a cell in your body or mine, and thereby create misery.
And it turns out the two structures look similar for a reason. The syringe on the right is made of a subset of the very same protein types found in the base of the flagellum on the left. Though the syringe is missing proteins found in the motor and therefore cannot produce rotary motion, it functions perfectly as an apparatus for transmitting disease.
So if we think about what it means to be irreducibly complex, the argument is that if you take away even one of these proteins, that the structure cannot function. And yet here is a structure that functions, that is missing several of the proteins, and yet here it is, a working, viable organelle of the bacterium. So indeed, this structure is not, in that sense, irreducibly complex.
To emphasize DeRosier's point, Miller arrived at court making an unusual fashion statement.
As an example of what irreducible complexity means, advocates of intelligent design like to point to a very common machine, the mousetrap. And the mousetrap is composed of 5 parts. It has a base plate, the catch, a spring, a little hammer that actually does the dirty work, and a bait holder. The mousetrap will not work if any one of these 5 parts are taken away. That's absolutely true. But remember the key notion of irreducible complexity, and that is that this whole machine is completely useless until all the parts are in place. Well, that turns out not to be true. And I'll give you an example. What I have right here is a mousetrap from which I've removed 2 of the 5 parts. I still have the base plate, the spring, and the hammer. Now, you can't catch any mice with this, so it's not a very good mousetrap. But it turns out that despite the missing parts, it makes a perfectly good, if somewhat inelegant, tie clip. And when we look at the favorite examples for irreducible complexity. And the bacterial flagellum is a perfect example. We find the molecular equivalent of my tie clip, which is we see parts of the machine missing 2, 3, 4, maybe even 20 parts, but still fulfilling a perfectly good purpose that could be favored by evolution. And that's why the irreducible complexity argument falls apart.
Just to call your attention to this idea that when we think about how a molecular machine works, what it does, that how the machine functions or the specific purpose that it plays for the organism can change over time. So just because something does one thing now doesn't mean that its predecessors always did that. Yes, you have a question?
Yeah. Would that be a case, like how you showed the two, like how one moved and the other didn't? Would that almost be a case of convergent evolution?
Okay, so the question was that if you look at the motor versus the syringe is stable, the motor turns around, and is that a case of convergent evolution or not? Because they have the same structure. So it's an interesting question in determining whether those two structures both arose independently or whether the motor evolved from the syringe. I mean, you would have to look at the specific components and understand their evolutionary history in order to determine whether those two structures arose independently or if one evolved from the other. I think that sort of the working hypothesis right now is that the syringe probably evolved first and then was added to become the flagellar motor. It's certainly, I'm not an expert in flagellum or these bacterial syringes, so I don't know if that's really current thinking right now, but that's one explanation for how that might have happened. Another one is you could have had a syringe evolve a flagellum and then lose components and become a syringe again. Or you could have two totally independently derived structures. And determining which of those scenarios is most plausible, you really have to get into the details about what the proteins are, what proteins are involved in that system, how they coordinate their efforts, what their evolutionary histories look like. So determining the particulars takes a lot of effort. So I want to now sort of shift focus a little bit, looking at the same question of complex molecular machines, and ask that as a question: are molecules and molecular systems machine-like or not? Because again, this is an analogy. We take a molecular system in nature and we say it has certain properties that seem very machine-like, but it may not necessarily be like a machine. So I'm going to show you guys another video on cell division. So this is the molecular basis of cell division, and this is built on our understanding of science, our understanding of how these cells divide. So I'm gonna play this, and I'm gonna ask you to watch it and just think about the way that cell division is being presented and how sort of machine-like it is.
DNA in each one of your cells is just 2 nanometers across, but around 30 to 40 million nanometers long. To keep DNA organized and regulate access to the genetic code, it is wrapped around structural proteins to form nucleosomes, which are then further organized into fibers called chromatin. The chromatin fibers are packaged into gigantic molecular structures called chromosomes. We are now pulling away from a chromosome, out through a nuclear pore, which is a gateway through the nuclear membrane. We are zooming back to reveal a cell about to undergo mitosis. The nuclear membrane breaks down, releasing the chromosomes. The chromosomes are arranged in the middle of the cell before being pulled apart with identical sets of DNA going to each side. The cell then splits down the middle, creating two new daughter cells.
So now we get the details of how that works.
This visualization isolates a single chromosome at mitosis. The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle, used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
All right, so that's it. If you've taken Intro to Biology, I mean, you probably learned about cell division. It's very well studied, and that's pretty much, probably what you'd get in a textbook. And it's accurate in terms of how we understand cell division to work. All the molecules in there are real molecules that do actual things. The chromosomes do separate and do things like that. So it is accurate from one point of view in that it conveys the information about how we understand cell division to work at the molecular level. And what did you think? Did it look pretty machine-like? It was ordered, it was regular, there were very few errors or mistakes. You didn't see molecules like flying off and bumping into things and doing that kind of stuff. If you saw all of that, it would be very difficult to understand the pattern. So that type of an illustration of cell division is for understanding the pattern. So we're removing a lot of the noise and all of the weird stuff that could be going on in the cell and emphasizing a certain particular outcome that we know actually occurs. And it looks very machine-like from that perspective. So I'm going to show, this one's really short. It's a stochastic model of what I think cell division would look like if all of the information and noise was just sort of thrown in there. And we saw what all of the molecules were doing. And it looks kind of like this. There's molecules falling off and getting back on and not being folded right and being recycled and just being... It's chaos. It doesn't make any sense. Now, I don't know if that's right. The reality is probably somewhere in between the two. But I think this idea that life is machine-like is another analogy. It's a way that we use to model the world to try to understand it, to try to communicate that understanding to other people in a meaningful way, and to try to then predict and reason about how the world works and how I can interact with it. And there'll be some aspects of that model that are good for certain things, and there'll be some aspects of that model that may not fit reality as closely as we might like. And so, again, I'm not here to like give you an answer about is the cell machine-like or not, but simply to raise the question that does thinking of it as machine-like make sense? Is it accurate representation of the way the cell actually is? And then, if not, what does the bias in emphasizing the machine-like quality of molecular systems, how does that bias the way we reason about evolutionary processes or their alternatives? And I want to kind of emphasize this point that I think I've been trying to talk about sort of tangentially throughout the hour, that all of these models are imperfect. That evolutionary theory is not the perfect explanation for everything that has gone on in life. That the machine-like nature of the cell is not perfect. That any model that we make of reality is not perfect. And so we really need to be careful when we're using our models of the world, our models of reality, to make decisions about what we want to do, what we want others to do, and things like that. And in addition to being really careful when we're trying to reason from models, we need to develop techniques and approaches to test our models continuously and make them better and better and better. And that's really what the scientific process does. So we're coming to the end. I was going to put up here, we face complex challenges. But I wanted to change it to you, because, you know, I'm kind of getting old, and I'm not going to be around for too much longer. But you guys are the next generation, and you really face some complex challenges. Things like, you know, global pollution and global warming, resurgence of disease, problems with trying to feed a growing population. You know, we have all sorts of problems with overcrowding and human population growth. And, you know, right now, for example, we're embroiled in, you know, brutal racially motivated violence in a lot of our societies. And these are real problems that you're going to face as you get older and move out into the world more and more and start taking over control of the human enterprise from old fogies who don't know anything like us. And these are hard. It's hard to solve these problems. We've been trying for hundreds of years to solve these problems, and we don't have perfect solutions. But what we need are you guys to step up and become real thinkers. You are poised in a situation where you have access to so much information. More information than even one generation, like exponentially more information than I had when I was in your position. You know, growing up in a technocratic democracy, you have access, I see two of you have computers right now, to vast storehouses of information. So getting the information isn't the problem. You can get information. The problem is sussing out the bullshit from the stuff that makes sense. And that's why we really need you guys to think. We need you to develop your capacity to critically evaluate that information and understand which of it makes sense, which of it doesn't make sense, and why. Because we've gotten really good at packaging nonsense in an easy-to-swallow package. And we do it intentionally and we do it unintentionally. And so there's a lot of stuff out there that's masquerading as good, solid information that's just not. And it's really hard to figure out which is which. And we need people to do this because we need people to make the right decisions to solve these problems in productive ways. So that's it from my 4 weeks. I'm going to thank you guys for showing up. I appreciate it. I think it's been a really fun and interesting class, and I hope it's challenged you guys to think not only about evolution, but about other important concepts in your lives. Thanks.